Hello everyone, I am Dr. Ritu Pandey, Assistant Professor in the Department of Botany, Hansraj College, University of Delhi. In this video lecture, I am going to discuss one of the most transformative innovations in molecular biology, polymerase chain reaction or PCR. While the name might sound complex, PCR is essentially a method of amplifying DNA. Think of it as a molecular photocopier that takes a tiny sample of DNA and multiplies it exponentially so we can analyze it in detail. What's truly fascinating is how PCR has become a keystone for so many fields such as medicine, agriculture, forensic science, and beyond. It's one of those techniques where you stop and think, what would science look like without it? In this lecture, I will cover the following aspects: introduction of PCR, application of PCR, requirements to perform PCR reaction, PCR workflow and cycle, advantages and disadvantages of PCR, and finally, the modifications of PCR techniques. What is PCR? Let me start with a little history. PCR was developed in 1983 by Kerry Mullis, who described the idea of replicating DNA sequences repeatedly in cycles. This simple yet brilliant concept won him the Nobel Prize in Chemistry in 1993. Why was it revolutionary? Because it took what was once nearly impossible, analyzing tiny amounts of DNA, and made it accessible, efficient, and scalable. Today, PCR is so embedded in research and diagnostics that it's hard to imagine molecular biology without it. Let's talk about why PCR is important and why it has revolutionized molecular biology. It allows us to amplify specific DNA sequences from even the tiniest amounts of genetic material. This capability has opened up endless possibilities across various fields. For example, in medicine, PCR is critical for diagnosing diseases. It helps detect infections caused by viruses or bacteria by amplifying their DNA or RNA, even when they are present in extremely small amounts. In forensic science, PCR is used to analyze tiny traces of DNA from crime scenes, helping to identify suspects or victims. In research, PCR enables us to study genes, identify mutations, and even understand the genetic basis of diseases. And in agriculture, it aids in the development of genetically modified crops that are more resilient to environmental stresses. So, in essence, PCR is important because it acts as a molecular magnifier, giving scientists and professionals the ability to detect, study, and manipulate DNA with incredible precision. Now, let's look at the requirements to perform PCR, the essential components that make this powerful technique work. First, we need a DNA template. This is the specific segment of DNA that we want to amplify. It could be from a patient sample, a crime scene, or even plant DNA material. Next, we need DNTPs, that is deoxynucleotide triphosphates, which are the building blocks of DNA, especially the four nucleotides DATP, TTTP, DGTP, and DCTP. These provide the raw materials to extend the DNA strands. We also require primers, which are short pieces of single-strand DNA. We use two primers, a forward primer and a reverse primer. They are designed to bind to the target DNA and provide the free 3' OH group necessary to initiate DNA polymerization. Then comes a thermostable DNA polymerase enzyme, such as Taq polymerase. This enzyme is key point of PCR because it can withstand the high temperature used during the denaturation step of PCR and synthesizes new DNA strands by adding nucleotides to the primer. Other polymerases like PFU and Wnt polymerase are used for specific purposes, especially when higher fidelity is required. Additionally, we need magnesium chloride, which acts as a cofactor for DNA polymerase enzyme, ensuring its optimal activity. Without magnesium ions, the polymerase cannot function effectively. To maintain the proper reaction environment, use a buffer solution. This ensures that the pH and ionic strength are suitable for the polymerase to work efficiently throughout the process. Finally, we need a PCR machine, also known as thermal cycler. This machine automates the cycling of temperatures required for denaturation, annealing, and extension, making the whole process fast and efficient. With all these components in place, we can set up the reaction and start amplifying DNA. Each of these elements play a crucial role in ensuring the reaction works properly. Now, let's get into the actual reaction of PCR, but I'll keep it simple. PCR works by taking advantage of the natural process of DNA replication, but it supercharges it in the lab using specific tools and control conditions. The process has three main steps: denaturation, annealing, and extension. During denaturation, the double-stranded DNA is heated to separate it into single strands. Think of it as unzipping the DNA molecule. In annealing, the short pieces of DNA called primers bind to their complementary sequence on the target DNA. Primers are like guides that tell the polymerase where to start copying. And finally, during extension, the new DNA strands are synthesized by adding nucleotides to the primer using a thermostable DNA polymerase enzyme. These steps are repeated in cycles, and with each cycle, the amount of DNA doubles. By the end of 25 to 30 cycle, you have millions or even billions of copies of the target DNA sequence. Now let's focus on the diagrammatic representation of the PCR cycle. This visual helps us understand the step-by-step process of how DNA is amplified during PCR. The cycle begins with the initial denaturation step. At this stage, the reaction mixture is heated to around 94 to 95 degrees Celsius for 1 to 5 minutes. This high temperature breaks the hydrogen bonds between the two strands of the DNA double helix, separating them into single strands. Next, we move to the first core step, denaturation, which happens at 94 degrees Celsius for about 30 seconds. This step ensures that the DNA remains single-stranded throughout the cycle. After denaturation, we have the annealing step. This is where the temperature is reduced to around 40 to 60 degrees Celsius, depending on the primers being used. At this temperature, the primers bind or anneal to their complementary sequences on the single-stranded DNA template. This step is crucial because primers define the starting point for DNA synthesis. The third key step is extension or elongation, which occurs at 72 degrees Celsius. This is the optimal temperature for the thermostable DNA polymerase, such as Taq polymerase, to add nucleotides to the primer and synthesize the new DNA strands. The duration of this step depends on the length of the DNA sequence being amplified, typically around 30 seconds to 2 minutes. At the end of the cycle, we have the final extension step held at 72 degrees Celsius for 1 to 10 minutes. This ensures that any incomplete DNA strands are fully synthesized. Finally, the reaction is held at 4 degrees Celsius to stabilize the amplified DNA product for storage or further analysis. The key to PCR is the repetition of denaturation, annealing, and extension steps. This is done in loop, typically for 25 to 35 cycles, and each cycle doubles the amount of DNA. As a result, you end up with millions or billions of copies of target DNA in just a few hours. This is how a typical PCR machine looks like. It is a compact device designed to automate the temperature changes required for the PCR process. While it may look simple on the outside, it's a sophisticated piece of equipment that plays a critical role in the lab. The machine contains a metal block with wells where reaction tubes are placed. These wells are designed to fit standard PCR tubes and ensure even heat distribution. What makes a PCR machine unique is its ability to rapidly cycle between different temperatures, following the steps of denaturation, annealing, and extension. This precise control of temperature is essential for the success of the reaction. Modern PCR machines come with features like programmable software, allowing users to set the number of cycles and customize the temperature and duration for each step. Some machines have advanced features for real-time monitoring, as in quantitative PCR. This machine is the backbone of every PCR experiment. Without it, the process would be extremely tedious and time-consuming if done manually. Now let's discuss the advantages and limitations of PCR, which highlight why this technique is so powerful yet not without challenges. Let's start with the advantages. Number 1. Sensitivity. PCR is incredibly sensitive, requiring only a small amount of starting DNA to generate millions of copies. This makes it invaluable in situations where DNA is limited, such as forensic sample or ancient DNA studies. PCR is very specific. That means the advantages is its specificity because it amplifies only the target DNA segment, guided by the primers designed for that sequence. This ensures accurate results. Speed. PCR is a fast technique. You can amplify DNA in just a few hours, making it an efficient tool for diagnostic and research. However, like any technique, PCR has its limitations. Knowledge limited. PCR requires prior knowledge of the DNA sequence to design specific primer. Without this information, it is impossible to target a specific region of DNA. Length limited. Amplifying long DNA fragments, usually over 10 kilobases, can be challenging. Standard PCR is better suited for shorter sequences. Error prone. DNA polymerases like Taq polymerase have a relatively higher error rate, approximately 1 error per 1,000 nucleotides. This can lead to inaccuracies, especially in applications requiring high fidelity. Repetitive sequences. PCR struggles with amplifying repetitive DNA regions due to issues with primer binding and elongation. While PCR is incredibly versatile, understanding these limitations is critical for designing experiments and interpreting results accurately. Now let's move on to the modification of PCR, which have expanded its application far beyond the basic technique. These modifications adapt PCR for specific needs and challenges, making it even more versatile in research, medicines, and diagnostics. Let's go through a few key modifications. RT-PCR or reverse transcriptase PCR. This is used to convert RNA into complementary DNA using the enzyme reverse transcriptase followed by PCR amplification. It is especially important for studying RNA virus like SARS-CoV-2 and analyzing their gene expression. QPCR or quantitative PCR or real-time PCR. In this method, the DNA amplification is monitored in real time using fluorescent dyes or probes. It allows us to quantify DNA and measure the initial amount of target DNA in a sample. This technique is widely used for pathogen detection, diagnostics, and gene expression studies. In multiplex PCR, we amplify multiple DNA targets in a single reaction by using multiple set of primers. It is efficient for detecting multiple pathogens or genetic markers simultaneously. In case of nested PCR, 2 rounds of PCR with inner and outer primer is performed to enhance sensitivity and specificity. It is useful when the target DNA is in very low abundance or when there is a high chance of non-specific amplification. In touchdown PCR, the annealing temperature is gradually decreased across cycles to enhance primer specificity. It helps to reduce non-specific binding and improve amplification efficiency. In hot start PCR, the reaction components are activated only after an initial high temperature step. This prevents non-specific amplification and primer-dimer formation during the reaction setup. Colony PCR. This is used to amplify DNA directly from bacterial colonies without requiring to purify the DNA. It is a quick method for screening bacterial clones. each of these modifications is tailored to overcome specific challenges or meet specialized requirements. For example, RT-PCR and qPCR are critical in modern diagnostics, while nested PCR improves accuracy when working with trace DNA. To wrap up, PCR isn't just a lab technique, it's a key that has unlocked countless doors in science. From understanding genetic disorders to tracking pandemics and even investigating evolutionary history, PCR has had a profound impact on humanity. As future researchers, you will likely encounter PCR in your work. Understanding its principles, strengths, and limitation will not only help you use it effectively, but also inspire you to innovate and push its boundaries further. Thank you.